A multimodal contrastive diffusion model for therapeutic peptide generation. Abstract. Therapeutic peptides represent a unique class of pharmaceutical agents crucial for the treatment of human diseases. Recently, deep generative models have exhibited remarkable potential for generating therapeutic peptides, but they only utilize sequence or structure information alone, which hinders the performance in generation. In this study, we propose a multimodal contrastive diffusion model, MMCED, fusing both sequence and structure modalities in a diffusion framework to co-generate novel peptide sequences and structures. Specifically, MMCED constructs the sequence modal and structure modal diffusion models, respectively, and devises a multimodal contrastive learning strategy with intercontrastive and intercontrastive in each diffusion time step, aiming to capture the consistency between two modalities and boost model performance. The intercontrastive aligns sequences and structures of peptides by maximizing the agreement of their embeddings, while the intercontrastive differentiates therapeutic and non-therapeutic peptides by maximizing the disagreement of their sequence/structure embeddings simultaneously. The extensive experiments demonstrate that MMCD performs better than other state-of-the-art deep generative methods in generating therapeutic peptides across various metrics, including antimicrobial/anticancer score, diversity, and peptide docking. Introduction. Therapeutic peptides, such as antimicrobial and anticancer peptides, are a unique class of pharmaceutical agents that comprise short chains of amino acids, exhibiting significant potential in treating complex human diseases. Traditionally, therapeutic peptides are discovered through a comprehensive screening of sequence spaces using phage/yeast display technologies or computational tools trained for scoring desired properties, Li et al. 2017, Li, Wang, and Ferguson 2018. However, the combinatorial space of possible peptides is vast, and only a small solution satisfies therapeutic requirements. Thus, such screening methods based on brute force can be time-consuming and costly. In recent years, deep generative models, DGMs, have demonstrated success in generating images, texts, proteins, and also gained popularity in peptides. DGMs explored a more expansive chemical space that affords the creation of structurally novel peptides by training neural networks to approximate the underlying distribution of observed or known ones, Juan, Canto-Jorgos, and Fuente 2022. For example, autoregression-based methods depicted peptide sequences as sentences composed of residue tokens, so that the problem can be solved by predicting residue arrangement via recurrent neural networks, RNN, Muller, Hiss, and Schneider 2018, Capecchi et al. 2021. Variational autoencoder, VAE, based methods generated new peptide sequences by sampling from the latent space learned through an encoder-decoder architecture, with or without therapeutic properties as conditional constraints. Gorbani et al. 2022, Simzak et al. 2023b. Generative Adversarial Network, GN. Based methods train the generator and discriminator using known data, which compete against each other to generate new peptides, Tux et al. 2020, Ort et al. 2021, Lin, and Lane 2022. Nowadays, diffusion models are prevalent in the generation of protein sequences and structures, owing to their superior capability in fitting distributions compared to prior techniques, Scher et al. 2023, Wu et al. 2022. Likewise, these advanced diffusion models can be extended to peptide generation and are expected to deliver favorable outcomes. Despite the commendable progress of efforts above, they focused on generating either sequences, i.e., residue arrangements, or structures, i.e., spatial coordinates of backbone atoms, ignoring that models fusing information from both modalities may outperform their unimodal counterparts. However, how to effectively integrate the multimodal information and capture their consistency in peptide generation is a major challenge. Additionally, compared with generation tasks for images, texts, and proteins that involve millions of labeled samples, public datasets for therapeutic peptides typically contain only thousands of sequence or structure profiles. induced by the high cost of in vitro screening. This limited amount of available data may result in overfitting, which confines generated outcomes within a restricted distribution, consequently compromising the model's generalization ability. ITY. How to fully leverage existing peptide data, such as therapeutic and non-therapeutic peptides, to enhance the generation performance could be regarded as another challenge. To address these challenges, we propose a multimodal contrastive diffusion model for therapeutic peptide generation, named MMCD. Specifically, we build a multimodal framework that integrates sequence modal and structure modal diffusion models for co-generating residue arrangements and backbone coordinates of peptides. To ensure consistency between the two modalities during the generation process, we bring in an intermodal contrastive learning (InterCL) strategy. InterCL aligns sequences and structures by maximizing the agreement between their embeddings derived from the same peptides at each diffusion time step. Meanwhile, to avoid the issue of inferior performance caused by limited therapeutic peptide data, we incorporate substantial known non-therapeutic peptides as data augmentations to devise an intramodal CL, InterCL. InterCL differentiates therapeutic and non-therapeutic peptides by maximizing the disagreement of their sequence/structure embeddings at each diffusion time step, driving the model to precisely fit the distribution of therapeutic peptides. Overall, the main contributions of this work are described as follows. We propose a multimodal diffusion model that integrates both sequence and structure information to cogenerate residue arrangements and backbone coordinates of therapeutic peptides, whereas previous works focused only on a single modality. We design the inter-intra-CL strategy at each diffusion time step, which aims to maximize the agreement between sequence and structure embeddings for aligning multimodal information, and maximize the disagreement between therapeutic and non-therapeutic peptides for boosting model generalization. Extensive experiments conducted on peptide datasets demonstrate that MMCD surpasses the current state-of-the-art baselines in generating therapeutic peptides, particularly in terms of antimicrobial/anticancer score, diversity, and pathogen docking. Related works. Diffusion model for protein generation. Diffusion models, Song and Ehrman 2019, Tripp et al. 2023, devote to learning the noise that adequately destroys the source data and iteratively remove noise from the prior distribution to generate new samples, which have emerged as cutting-edge methods for numerous generation tasks, especially in proteins, Wu et al. 2022, Chow et al. 2023. For example, Liu et al. 2023 proposed a textual conditionally guided diffusion model for sequence generation. Hoochboom et al. 2022 introduced ProtDiff with an E3 equivariant graph neural network to learn a diverse distribution over backbone coordinates of structures. Luo et al. 2022 considered both the position and orientation of antibody residues, achieving an equivariant diffusion model for sequence structure cogeneration. Despite their success, the fusion of both sequence and structure modalities in diffusion models has not been comprehensively investigated, and their potential for peptide generation remains unexplored. To fill this gap, we implement a peptide-oriented diffusion model capable of sequence structure cogeneration and multimodal data fusion. Contrastive learning. Being popular in self-supervised learning, contrastive learning (CL) allows models to learn the knowledge behind data without explicit labels. Xia et al. 2022, Zhu et al. 2023. It aims to bring an anchor, i.e., data sample, closer to a positive/similar instance and away from many negative/dissimilar instances by optimizing their mutual information in the embedding space. Strategies to yield the positive and negative pairs often dominate the model performance. For example, Yuan et al. 2021 proposed a multimodal CL to align text and image data, which encourages the agreement of corresponding text-image pairs, positive, to be greater than those of all non-corresponding pairs, negative. Wu, Liu, and Dong, 2022, designed a CL framework that makes full use of semantic relations among text samples via efficient positive and negative sampling strategies to mitigate data sparsity for short text modeling. Zheng et al. 2023b augmented the protein structures using different conformers and maximized the agreement/disagreement between the learned embeddings of same/different proteins, aiming to learn more discriminative representations. However, these CL strategies have yet to be extended to peptide-related studies. Therefore, we devised the novel CL strategy in peptide generation, which serves as an auxiliary objective to enforce sequence structure alignment and boost model performance. Methodology. In this section, we formulate the peptide cogeneration problem for sequence and structure. Subsequently, we elaborately enumerate the components of our method MMCD, including the diffusion model for peptide generation and the multimodal contrastive learning strategy. The overview of MMCD is illustrated in Figure 1. Problem formulation. A peptide with N residues, amino acids, can be represented as a sequence structure tuple, denoted as X equals SC. S equals S subscript I equals 1 to the power n, stands for the sequence with S subscript I as an element of A, C, D, E, F, G, H, I, K, L, M, N, P, Q, R, S, T, V, W, Y as the type of the I, TH residue. And C equals C subscript I equals 1 to the power n, stands for the structure with C subscript I as an element of R to the power 3 asterisk operator 4 as Cartesian coordinates of the I, TH residue, involving 4 backbone atoms, N minus C subscript alpha minus C minus O. Our goal is to model the joint distribution of X based on the known peptide data so that sequences, i.e., residue types, and structures, i.e., residue coordinates, of new peptides can be cogenerated by sampling the distribution. Diffusion model for peptide generation. The diffusion model defines the Markov chains of processes in which latent variables are encoded by a forward diffusion process and decoded by a reverse generative process. Figure 1. Overview of the MMCD. MMCD consists of a diffusion model for the peptide sequence structure cogeneration and multimodal contrastive learning (CL). The diffusion model involves a forward process, Q divides, for adding noise, and a reverse process, P divides, for denoising at each time step T. The reverse process utilizes a transformer encoder, or EGNN, to extract embeddings from sequences, S, or structures, C, and a sequence, or structure, based MLP to map embeddings to the marginal distribution, or Gaussian, noise. The multimodal CL includes an inter-CL and an intra-CL, which aims to align sequence and structure embeddings and differentiate therapeutic and non-therapeutic peptide embeddings. Soldikstein et al. 2015. Let X to the power 0 equals S to the power 0, C to the power 0, denotes the ground truth peptide, and X to the power T equals S to the power T, C to the power T 4, T equals 1 T, to be the latent variable at time step T. The peptide generation can be modeled as an evolving thermodynamic system, where the forward process, Q, X to the power T divides X to the power T minus 1, gradually injects small noise to the data, X to the power 0, until reaching a random noise distribution at time step T, and the reverse process, P subscript theta, X to the power T minus 1 divides X to the power T, with learnable parameters, theta, learns to denoise the latent variable, X to the power T, towards the data distribution. Diffusion for peptide sequence. Following Anand and Occam, 2022, we treat residue types as categorical data and apply discrete diffusion to sequences, where each residue type is characterized using one hot encoding with 20 types. For the forward process, we add noise to residue types using the transition matrices with the marginal distribution, Austin et al. 2021, Vignac et al. 2023, see details in Appendix A. For the reverse process, the diffusion trajectory is parameterized by the probability, Q, S to the power T minus 1 divides S to the power T, S to the power 0, and a network, p caret subscript theta, is defined to predict the probability of s to the power 0, Austin et al. 2021, that is. The probability of a past state given a current state is a product of individual probabilities and a probability of an initial state, where s subscript I to the power t denotes the one-hot feature for the th residue in the sequence, s at time step t, and s caret to the power 0 is the predicted probability of s to the power 0. In this work, we design the p caret subscript theta as follows. The estimated probability of a state sequence is the product of softmax probabilities for each state, where h subscript I to the power t is the input feature of residue, I, with the diffusion noise at time, t. The initialization of h subscript I to the power t is provided in Appendix A. F subscript S is a hybrid neural network to predict the noise of residue types from the marginal distribution, and then the noise would be removed to compute the probability of S caret subscript I to the power 0. Softmax is applied over all residue types. Here, we implement F subscript S with a transformer encoder and an MLP. The former learns contextual embeddings of residues from the sequence, while the latter maps these embeddings to the noises of residue types. The learned sequence embedding, defined as S, involves downstream contrastive learning strategies. Diffusion for peptide structure. As the coordinates of atoms are continuous variables in the 3D space, the forward process can be defined by adding Gaussian noise to atom coordinates, Ho, Jane, and Abiel 2020. See details in Appendix A. Following Tripp et al. 2023, the reverse process can be defined as: the probability of a variable is a normal distribution with its mean defined by a separate equation, where C subscript I refers to coordinates of the th residue in the structure. C, beta, is the noise rate, formally alpha to the power t equals 1 minus beta to the power t, alpha to the power t equals pi tau equals 1 ton. 1 minus beta to the power tau, the network, epsilon subscript theta, is used to gradually recover the structural data by predicting the Gaussian noise. In this work, we design the epsilon subscript theta as follows. The epsilon of theta of c at time t equals the f of c of r at time t and h at time t, where r subscript I represents the coordinates of residue, I, h subscript I is the residue feature, and f subscript c is a hybrid neural network for predicting Gaussian noises at time step t. Similar to sequence diffusion, we implement f subscript c with an equivariant graph neural network, EGNN, Satoras, Hujboom, and Welling 2021, and an MLP. The former learns spatial embeddings of residues from the structure, formalized as a 3D graph, while the latter maps these embeddings to Gaussian noises. The learned structure embedding, defined as C, also involves downstream contrastive learning strategies. Diffusion objective. Following previous work, we decompose the objective of the peptide diffusion process into sequence loss and structure loss. For the sequence loss L subscript S to the power T, we aim to minimize the cross-entropy, C, loss between the actual and predicted residue types at time step T. The loss function is the average cross-entropy between true and predicted labels for a set of samples. For the structure loss L subscript C to the power T, the objective is to calculate the mean squared error, MSE, between the predicted noise, epsilon subscript theta, and standard Gaussian noise, epsilon, at time step t. The loss function L underscore C caret t is the average squared difference between epsilon underscore I and epsilon underscore theta. Multi-modal contrastive learning strategy. When multiple modal data, e.g., sequence and structure, coexist, it becomes imperative to capture their consistency to reduce the heterogeneous differences between modalities, allowing them to be better fused in generation tasks. Mutual information, MI, is a straightforward solution to measure the nonlinear dependency, consistency, between variables. Thus, maximizing MI between modalities can force them to align and share more crucial information. Along this line, we bring in contrastive learning, CL, to align sequences and structures by maximizing their MI in the embedding space. Specifically, we devise CL strategies for each diffusion time step T as follows. Inter-CL. For a peptide, we define its sequence as the anchor, its structure as the positive instance, and the structures of other peptides in a mini-batch as the negative instances. Then, we maximize the MI of positive pair, anchor and positive instance, while minimizing the MI of negative pairs, anchor and negative instances, based on embeddings learned from the networks, p caret subscript theta and epsilon subscript theta. Further, we establish a dual contrast where the structure acts as an anchor and sequences are instances. The objective is to minimize the following InfoNCE-based loss function. The interclass loss at time t is the negative average of two log ratios, where S subscript I / C subscript I is the sequence / structure embeddings of I, th peptide in the mini-batch, E is the cosine similarity function with the temperature coefficient to measure the MI score between two variables, M is the size of a mini-batch. In addition, the used diffusion model can only remember confined generation patterns if therapeutic peptide data for training is limited, which may lead to inferior generalization towards novel peptides. To alleviate this issue, we introduce contrastive learning to boost the generative capacity of networks, p caret subscript theta and epsilon subscript theta, by enriching the supervised signals. However, it is unwise to construct positive instances by performing data augmentations on therapeutic peptides, as even minor perturbations may lead to significant functional changes, Yadav, Kumar, and Singh 2022. Hence, our focus lies on employing effective strategies for selecting negative instances. In this regard, we collect non-therapeutic peptides from public databases to treat them as negative instances and maximize the disagreement between embeddings of therapeutic and non-therapeutic peptides. In detail, we devise an intra-CL strategy for each diffusion time step T as follows. Intra-CL. In a mini-batch, we define the sequence of a therapeutic peptide, I, as the anchor, and the sequence of another therapeutic peptide, J, as the positive instance, while the sequences of non-therapeutic peptides, K, are regarded as negative instances. Similar to inter-CL, we then maximize / minimize the MI of positive / negative pairs. And we also establish a structure-oriented contrast by using structures of therapeutic and non-therapeutic peptides to construct the anchor, positive, and negative instances. The objective is to minimize the following loss function. The interclass loss at time t is a sum over pairs of samples, weighted by whether they share the same label, where Y subscript I represents the class of peptide I, i.e., therapeutic or non-therapeutic. 1 subscript Y subscript I equals Y subscript J, and 1 subscript Y subscript I is not equal to Y subscript K stand for the indicator functions, where the output is 1 if Y subscript I equals Y subscript J, peptides I and J belong to the same class, or Y subscript I is not equal to Y subscript K. The types of peptides I and K are different, otherwise the output is 0. The indicator function filters therapeutic and non-therapeutic peptides from the data for creating positive and negative pairs. Table 1. Results for the sequence generation. Asterisk represents that the method relies on domain-specific biological knowledge. Dash represents that the method is unsuitable for the current task. For example, AMPGAN and HydRAMP are only designed for the AMP generation. Table 1. Results for the sequence generation. Table 2. Results for the structure generation. The reason behind the design of intra-CL is intuitive. First, the non-therapeutic class naturally implies opposite information against the therapeutic class, and hence it makes the model more discriminative. Second, the fashion to maximize the disagreement between classes 1 can induce biases in the embedding distribution of therapeutic peptides, identifying more potential generation space, and 2 can explicitly reinforce embedding class correspondences during diffusion, maintaining high generation fidelity. Further analysis is detailed in the ablation study. Model training. The ultimate objective function is the sum of the diffusion process for sequence and structure generation, along with the CL tasks for intra-CL and inter-CL. L subscript total equals E subscript T tilde operator uniform 1. T alpha L subscript S to the power T plus L subscript C to the power T plus 1 minus alpha L subscript intra to the power T plus L subscript inter to the power T, where alpha represents a hyperparameter to balance the contributions of different tasks. The uniform 1. T shows the uniform distribution for the diffusion time steps. The implementation details of MMCED and the sampling process of peptide generation can be found in Appendix A. Experiments. Experimental setups. Datasets. Following previous studies, T-Fan et al. 2022, Zhang et al. 2023a, we collected therapeutic peptide data from public databases containing two biological types, i.e., antimicrobial peptides (AMP) and anticancer peptides (ACP). Among these collected peptides, a portion of them only have 1D sequence information without 3D structure information. Then, we applied Rosetta-based computational tools, Chauthuri, Liskov, and Gray 2010, to predict the missing structures based on their sequences. Finally, we compiled two datasets, one containing 20,129 antimicrobial peptides and the other containing 4,381 anticancer peptides. In addition, we paired an equal number of labeled non-therapeutic peptides collected from public databases with each of the two datasets exclusively for the contrastive learning task. Baselines. We compared our method with the following advanced methods for peptide generation at sequence and structure levels. For the sequence generation, the autoregression-based method LSD-MRNN, Muller, Hiss, and Schneider 2018, the GAN-based method AMPGAN, and the VE-based methods including WAPSO and HydRAMP are listed as baselines. For the structure generation, we took APTIST as a baseline, which combines the neural network and simulated annealing algorithm for structure prediction. Moreover, we extended diffusion-based methods for protein generation to peptides. The diffusion-based methods for structure generation, e.g., folding diff and prot-diff, and the sequence structure codesign, e.g., diffab and simdiff, are considered for the comparison separately in the sequence and structure generation. Evaluation protocol. Here, we required each model, Rs and baselines, to generate 1,000 new peptides and then evaluated the quality of generated peptides with the following metrics. For the sequence, similarity score is used to Figure 2, A, the sample ratio under different sequence lengths in the AMP dataset, where the red line is the average ratio. B, the similarity and RMSD scores of MMCED and baselines across different sequence lengths. Quantify how closely the generated sequences match existing ones, with a lower score indicating higher novelty. Instability score, Muller et al. 2017, indicates the degree of peptide instability. Antimicrobial/anticancer score evaluates the probability of peptides having therapeutic properties. For the structure, Ramachandran score accesses the reliability of peptide structures. RMSD score measures the structural similarity between generated and existing peptides, with a lower score indicating higher authenticity. Docking score. Flores-Castillo et al. 2020 evaluates the binding degree of antimicrobial peptides to bacterial membrane proteins, PED-BID, 6Mi7. We only reported the average metrics over all generated peptides for each method in the experimental results. Detailed information about the datasets, baselines, metrics, and implementations can be found in Appendix B. Our code, data, and appendix are available on GitHub, https://github.com/wiki4811/mmcd. Experimental results. Performance comparison. In the results of sequence generation under two datasets, as shown in Table 1, MMCED exhibited lower similarity and instability scores than all baselines, suggesting its good generalization ability in generating diverse and stable peptides. Meanwhile, MMCD surpassed all baselines with higher antimicrobial and anti-cancer scores across AMP and ACP datasets, highlighting its strong potential for generating therapeutic peptides. Beyond that, we noticed that diffusion-based baselines, e.g., SIMDiff, DiffAB, exhibit higher stability and diversity, but lower therapeutic scores compared to baselines that incorporate biological knowledge, e.g., AMPGAN, HYDRAMP, WAPSO, details in Appendix B. By contrast, MMCD introduced biological knowledge into the diffusion model by designing the contrastive learning of therapeutic and non-therapeutic peptides, thereby delivering optimality across various metrics. For the results of structure generation, as shown in Table 2, MMCD also outperformed all the baselines and exceeded the best baselines, DiffAB and SIMDiff, by 23.3% and 12.9%. In RMSD scores, 10.2% and 5.6% in Ramachandran scores, and 7.4% and 2.7% in docking scores for AMP dataset. The higher Ramachandran score and lower RMSD score of MMCD underline the reliability of our generated peptide structures. Especially in peptide docking, we found that MMCD shows the best docking score compared with baselines, which indicates great binding interactions with the target protein. Overall, MMCD is superior to all baselines in both sequence and structure generation of peptides, and its impressive generative ability holds great promise to yield high-quality therapeutic peptides. Performance on different sequence lengths. In our dataset, sequence lengths of different peptides exhibited substantial variation, with the number of residues ranging from 5 to 50, Figure 2a. We required models to generate 20 new peptides, sequences, or structures at each sequence length. Note that two methods, AMPGAN and HYDRAMP, were excluded from the comparison because they cannot generate peptides with fixed lengths. From the generated results on the AMP dataset, Figure 2b, MMCD exceeded the baselines in terms of similarity and RMSD scores at each sequence length. With the increasing sequence lengths, there is a general trend of increased similarity and RMSD scores across all methods. One possible reason for this trend is that designing longer peptides becomes more complex, given the more prominent search space involved. Additionally, the scarcity of long-length peptides poses challenges in accurately estimating the similarity between generated and known peptides. In summary, these observations supported that MMCD excels at generating diverse peptides across different lengths, especially shorter ones. Ablation study. To investigate the necessity of each module in MMCD, we conducted several comparisons between MMCD with its variants. 1. MMCD, without interCL, that removes the interCL task. 2. MMCD, without interCL, that removes the intraCL task. And 3. MMCD, without interCL and interCL, that removes both interCL and intraCL tasks. The comparisons were operated on both AMP and ACP datasets, and the results are shown in Table 3 and Appendix Table 1. When the interCL was removed, without interCL, we observed a decline in all metrics for peptide sequence and structure generation, implying the importance of aligning two modalities via CL. The variant, without interCL, results signified that using the CL to differentiate therapeutic and non-therapeutic peptides contributes to the generation. As expected, the performance of MMCD dropped significantly after removing. Table 3. Ablation study on the sequence-level generation task. Figure 3a. The T-SNE for structure and sequence embeddings of therapeutic peptides, AMP data, obtained from MMCD, without interCL, and MMCD. B. The T-SNE for embeddings, including structures and sequences, of therapeutic, AMP, and non-therapeutic, non-AMP, peptides obtained from MMCD, without interCL, and MMCD. Both interCL and interCL, without interCL and interCL. To better understand the strengths of interCL and intraCL, we performed the T-SNE visualization using the learned embeddings of peptides on the AMP dataset. As illustrated in Figure 3a, Interciel effectively promoted the alignment of sequence and structure embeddings, facilitating the shared crucial information, dashed circle, to be captured during diffusion. The T-SNE of Interciel, Figure 3b, also revealed that it better distinguished therapeutic peptides from non-therapeutic ones in the embedding distribution. And the resulting distribution bias may identify more potential generation space, thus leading to higher quality and diversity of therapeutic peptides generated by MMCD. Overall, MMCD with all the modules fulfilled superior performance, and removing any modules will diminish its generation power. Peptide docking analysis. To test the validity of generated peptide structures, we conducted a molecular docking simulation. Here, a peptide was randomly selected from the AMP dataset as the reference, and the methods, Figure 4, were employed to generate corresponding structures based on the sequence of the reference peptide. See details in Appendix C. The lipopolysaccharide on the outer membrane of bacteria, Li, Orlando, and Liao 2019, was selected as the target protein for molecular docking. Then, we extracted the residues within A5 proximity between peptides, i.e., the reference and generated structures, and the active pocket of target protein in docking complexes to visualize their binding interactions. Of these docking results, all methods yielded a new structure capable of binding to the target protein, and our method exhibited the highest docking scores and displayed binding residues most similar to the reference structure. This prominent result underscored the reliability and therapeutic potential of our method for peptide generation. Figure 4. Docking analysis. Interactive visualization between target protein and peptides of the reference and generated structures by MMCED and baselines. Thick lines represent the residues of peptides, and the thin lines show the binding residues for protein-peptide complexes. Conclusion. In this work, we propose a multimodal contrastive diffusion model for the cogeneration of peptide sequences and structures, named MMCD. MMCD is dedicated to leveraging a multimodal contrastive learning strategy to capture consensus-related and difference-related information behind the sequences/structures and therapeutic/non-therapeutic peptides, enhancing the diffusion model to generate high-quality therapeutic peptides. The experimental results unequivocally demonstrate the capability of our method in co-generating peptide sequence and structure, surpassing state-of-the-art baseline methods with advantageous performance. Acknowledgements. This work was supported by the National Natural Science Foundation of China 6237-2204, 6207-2206, 6177-2381, 6210-2158. Washing Agricultural University Scientific and Technological Self-Innovation Foundation, Fundamental Research Funds for the Central Universities, 2662021JC008, 2662022JC004. The funders have no role in study design, data collection, data analysis, data interpretation, or writing of the manuscript.